Hi everyone, good evening. So we're going to continue talking about the material that the professor gave in class for the next exam, which is April 2nd. And this is going to be on the amino acid slides that the professor gave. So amino acids, anime acids, whatever you want to call them is a funny, is a funny pun. Anyways, food industry uses. You're going to have a bunch of flavor enhancers associated with amino acids. You of course have monosodium glutamate, or just glutamate. This is going to be that umami, kind of a mixture of salty, sweet, and just a meaty taste. Very it has this like depth to it. I think it's like the flavor of depth, so it just enhances. Okay, aspartate and alanine, those are going to be for fruit juice. Cysteine, you could enhance bread quality because it will reduce the bonds in the bread, and you could also have it as an antioxidant in fruit juice. Glycine is going to be used in sweeteners. Okay, let's talk about sweeteners in general, so not just what enhances them, but now we're just going to the sweetener itself. So phenylalanine as well as aspartic acid, these are going to make aspartame. Some brands are Equal and NutraSweet. Alright, essential amino acids. So these essential amino acids are the ones that humans can't make. We can't make 8 out of the 20 amino acids. Some examples are lysine, methionine, threonine, and tryptophan. We can't make these as examples. There are 3 approaches to amino acid production. You could extract from the protein hydrolysates. So you could get cysteine, leucine, asparagine, tyrosine. You could also do it by chemical synthesis. This is where you get glycine, alanine, methionine, and tryptophan. Or you could also source them from microbes, and since this is an applied micro, that's the one I think we're going to have to spend time on. Alrighty. 3 approaches in microbial production. You could use direct fermentation. You're going to be using a cheap molasses, starch hydrolysates. Then you could also convert the inexpensive intermediates into something. So glycine could actually be converted into serine. Enzymes or immobilized cells could also help with the production of amino acids from microbes. You're going to be dealing with these stereospecific amino acid acylases from Aspergillus oryzae. So an acylase, and you're going to split chemically synthesized DL amino acid that's been acetylated. You're going to just basically sort them using this acylase or Aspergillus oryzae. Okay, the biochem aspect of amino acids, again, you get the precursors as well as the amino acids can be formed in these pathways from those precursors. Like the glycolysis and TCA. So those are like, they're derived from the catabolism of glucose because, again, remember, glucose, sugars, carbs are kind of the backbone of everything. Okay, now, naturally occurring glutamate. So again, talking about like MSG, the king of flavor, as one of my favorite YouTube chefs / comedians always says, it's the king of flavor. And yeah, hiya. Okay, so tomato, you're going to have 0.5 grams, almonds 6.8, soy sauce 1.6. We don't need to know these numbers, just need to know some of these things. Spirulina has the highest out of the plants, and then it's not even a plant, that's from an algae. One that is the highest is soy protein isolate. Sorry about that. Read the chart incorrectly. Alright, on the animal side, Parmesan cheese has a lot of glutamate. Beef loin has only 6. Whey protein, that's going to be 14, and then bacon is 5.6. Again, it's the flavor of kind of like this coming together of sweet and salty, and then like weight. There's like a depth again. Right. The microorganism that makes glutamate is Corneobacterium glutanicum. You could kind of guess in the name there. You need biotin because it's naturally oxytrophic for biotin. This gram-positive species is naturally oxytrophic for biotin, so you need to supply the biotin to this organism. So while you do need to supply the biotin, you also need to take into account that you need this biotin starvation. You don't want to give it too much biotin. You want to just supply it suboptimally, so a little bit less than it actually needs. Biotin serves as this prosthetic group. It's a non-protein group on the acetyl-CoA carboxylase. This is the first step in fatty acid synthesis. And the insufficiency in fatty acid leads to the increased membrane permeability. This is why biotin starvation leads to an increase in glutamate. When you're starving biotin, you're not going to be making this fatty acid, so therefore you're going to have the membrane more permeable, and then so therefore more glutamate is actually going to be released. Insufficiency in fatty acid leads to increased permeability. The insufficiency in fatty acid comes from biotin being limited. Alright, the biosynthesis of glutamate is this chart here. Isocitrate to alpha-ketoglutarate, that's always going to be tied in the microbe to NADP and NADPH. Again, reduction, oxidation there. L-glutamate will come from NH4 plus alpha-ketoglutarate. So it's kind of all connected, you know. Alpha-ketoglutarate is the key precursor from the TCA cycle. It's converted into L-glutamate by reductive amination. So this is known as reductive amination. With, of course, free ammonia. Okay, metabolic flux analysis. This is difficult to do in an intact cell. However, you're going to be seeing the initial studies done in cell extracts. This is going to be concerning the flow of the pathway. So, metabolic control analysis, this is when the flux alterations are calculated from enzyme levels and the amount of carbons that go through. That's what you're basically concerning yourself with. Metabolic control analysis only works when the segment of the pathway is not regulated by an intermediate or an end product. Metabolic balancing, this is how fast the glucose is used. So the measure of the consumption of carbon and energy source, as well as the excretion of products such as lactate, this acetate, and amino acids. You're going to combine this with isotopic labeling, and you're going to have a model with these flux figures for each step of metabolism. So flux in pathways, so exponentially growing cells, the flux to glutamate is 0. However, when you have cells growing in limited biotin, this will have this effect where the influx of glucose happens less than 50% of exponentially growing cells, and you're going to also have the contribution of the pentose phosphate pathway minimalized, and half of the alpha-ketoglutarate is therefore going to become glutamate once we shut down the pentose phosphate. Now 50% of the alpha-ketoglutarate is siphoned off to glutamate. The feedstock for the Corneobacterium glutanicum will be simple glucose, glucose sucrose. sugarcane and sugar beet molasses could also be used. So these molasses from the sugar beet and sugar cane, they actually contain some of these they contained biotin. Oh wait, did I read that correctly? Let me think. Yeah, small amount of biotin is added. Level is dependent on the carbon source. Sorry about that. I had a brain fart. Okay, more conditions. Oxygen level, that's the main thing. Too high, it will make alpha ketoglutarate. Too low of an oxygen level will make succinate as well as lactic. pH at 7.8, temperature is 32 initially, then you increase to 38 after about 14 hours. The fermentation itself ends at 35. Alright, let's talk about recovery. Cells are separated by centrifugation. We've seen that already. Glutamate is obtained by precipitation and electrodialysis, ion exchange, or solvent extraction. That's how you obtain the glutamate. L-lysine is an essential amino acid, therefore we can't make it. It occurs in low concentrations in plant proteins. It's only therefore made by microbes. This is interesting. Adults need 38 milligrams per kilogram per body weight per day, and this lysine is also used in animal feeds where limited. The amino acid in pigs, and you're playing around with those forces. And let me think what else I wanted to say here. Supplementation also allows for low-cost corn to be used. Okay, so the microorganism for lysine will be Corneobacterium glutanicum again, or Brevibacterium flavum. Mutants are auxotrophs for combinations of homoserine, leucine, threonine are used. So we're going to use mutants of homoserine, leucine, threonine. Okay. I showed the strains of the bacteria, the Brevibacterium flavum or the Corneobacterium glutanicum. They are AEC resistant. So AEC, this means it's insensitive to feedback inhibition. So whatever happens, it's gonna still go. It's not ever going to be inhibited. AEC is a lysine analog. Okay, the feedstock again. This is going to be the sugar, cheap stuff, cane molasses. And you also need some nitrogens, of course, new ones, urea, ammonium, salt gaseous, or liquid ammonia. And then you also have the soy protein hydrolysate. Homoserine or threonine and methionine must be added to suboptimal concentrations. And then biotin is added if beet molasses specifically, because the beet molasses is deficient. Fermentation conditions, same stuff basically, 60 hours. Let me see. I guess I didn't write this part down. Oh, here it is. 28 degrees Celsius, pH 7, 60 hours. And that's all on amino acids. Please do something nice for someone, and have a good one.